Hello, biologists. Welcome back to our class. We are starting chapter 13 now, where we are going to explore the cytoskeleton within our cells. The cytoskeleton is a network of proteins within the cell. The interior of our cells are very highly structured, and a lot of that is due to the cytoskeleton. The cytoskeleton is a network of different proteins, different filaments and tubules that are connected to each other and extends throughout the cytosol. It plays roles in the cell being able to move and in the cell being able to divide. And to be able to do these different things, the cytoskeleton needs to be dynamic and changeable. It is not just this is a cytoskeleton forever and ever, but it alters, it'll add proteins and take proteins and change those proteins to accommodate the needs of the cell. There are three major structural elements in a cytoskeleton. These are microtubules, that's what we see here in red, microfilaments, and intermediate filaments. Microtubules are hollow tubes. They are about 13 protofilaments that are composing these tubes. A monomer would be called alpha tubulin or beta tubulin. They have a polarity. They will use GTP to form. And their functions, there are cytosolic microtubules that help organize and maintain animal cell shape and polarity. They help chromosomes to move, especially important during mitosis. And they are in charge of intracellular transport and trafficking, helping organelles move from one area of the cell to another. There are also axonal microtubules that are involved in cell motility. Next are microfilaments. These are two chains that are intertwined around each other, woven together. They are much thinner in diameter. Their monomers are G-actin, and they are also polar. They use ATP as a nucleotide substrate. And microfilaments are involved in muscle contraction, cell locomotion, cytoplasmic streaming, cytokinesis, maintaining the shape of an animal cell, and trafficking and intracellular transport within the cell. Then intermediate filaments are 8 protofilaments that are joined end to end. They have staggered overlaps. You can see they begin and end at staggered edges. There are 6 classes of proteins that we'll explore later this chapter. They don't have a known polarity. They do not have a nucleotide substrate. We skipped over diameter. You can see they're called intermediate because they're intermediate in size. They're not as small as the microfilaments, but they're not as large as the microtubules. They're intermediate here. Okay, their functions are structural support, maintaining the shape of an animal, forming the nuclear lamina, which lines the nuclear membrane, and scaffolding, strengthening nerve cell axons, or neurofilament proteins, and keeping myofibrils in register. Bacteria have cytoskeletons that are similar to eukaryotes. They are not quite the same. So they have polymer systems that function similar. They have an actin-like MRAB protein that helps DNA segregation and maintain cell shape. MRAB also associates with proteins that form the cell wall. They have a tubulin-like FITZ protein that is involved in regulating cell division. And crescentin is a regulator of cell shape. So you can see they do similar functions, provide similar benefits to prokaryotic cells, but they are not the same proteins as we see in our eukaryotic cells. The cytoskeleton can be dynamically assembled and disassembled. It can be put together, it can be taken apart, it can change its shape to fit in the different requirements of the cell. So microfilaments are components of muscle fibrils. Microtubules are structural filaments of cilia and flagella. These are structures that you can actually see with a microscope if you were to look at it. And the cytoskeleton is dynamically assembled and disassembled and includes remarkably elaborate structures. Okay, so for the remainder of this chapter, we are going to explore those different types of proteins that are in our cytoskeleton in a little bit more depth. So I'll see you soon. Let me know if you have any questions. Bye.